Welcome back friends. Welcome to another video tutorial from Shomu's Biology. And in this video lecture, I'll be talking about Ramachandran plot. So before understanding Ramachandran plot, you need to know about the torsional angles. Okay. So if you know torsional angles, then it's fine. If you don't know, then please watch my video on torsional angles or dihedral angles. Okay. And we'll talk about the dihedral angles, and we'll also talk about the importance of dihedral angles in maintaining the protein structure, especially the protein secondary structure. And we'll also see and talk about Ramachandran plot, which gives us the idea about what kind of torsional angle will ultimately fit to what type of secondary structure of the proteins. So let's see the relation between torsional angles and the Ramachandran plot and protein structure. So let's begin with this. The idea of torsional angles or dihedral angles originated from a three-dimensional space angle idea. Now what does that mean? If you look at here, the whole idea of torsional angles we'll talk about for the proteins or the amino acid chain, which is also known as polypeptide. So if you look at this polypeptide, it contains 1, 2, 3 different amino acid chains linked with one another. Now if you look at here, there is an N terminal of the amino acid. This is the N terminal, and this is the C terminal of the amino acid. Okay. So these are the two different terminals that we depict here. Now in this case, the peptide bonds are this one, CONH. This will be known as the peptide bond, because these are separate amino acids linked with this bond here. Now one thing you should look very carefully here is that this nitrogen always carry lone pair of electron. As a result, in a resonance property, this lone pair of electron can form another bond between this carbonyl carbon and the nitrogen. So as they form this bond, a partial side bond between carbonyl carbon and nitrogen. Previously there was one bond, so now it kind of behave like two bonds in between the carbonyl carbon and the nitrogen that are involved in a peptide linkage. Now that is called a peptide bond. And as I told you, if you look at here, that peptide bond contains two bond characteristics. So it's a partially double bond character of the peptide bond. It acts like a partially double bonded bond. Now in this case, this partial double bonds that we can see, and here you see the carbonyl, this is the alpha carbon that are placed in between. Now as a result of this double bond, partial double bond nature of the peptide linkage, this amino acids or all those molecules cannot rotate around this bond. It will be unable to rotate around this partial double bond character. Okay, because it's kind of a double bond feature, it is restricted to rotate. So all those amino acids this side and this side cannot rotate. On the other hand, if you look here, there are two other bonds that you also find. One is this carbonyl carbon and C alpha carbon, the bond between them, this one. Another one is the bond between the carbonyl carbon, the nitrogen and the carbonyl carbon. So these are the alpha carbon, sorry. So these are the two bonds that we can see. The alpha carbon in the middle, in the left you see the bond between nitrogen and the alpha carbon. This is one type of bond. And the other bond is the bond between carbonyl carbon and the alpha carbon. Those are two different bonds. And those bonds have specific names. Here the bond between the N-C alpha is known as the Phi bond, and the bond between C alpha C is known as the Sai bond. So let me write them here. This is Phi bond, this is Sai bond. Two different bonds are there. Now this is up to the link and only the peptide bond. But now you'll see how we talk about angles in here. The simple thing I can tell you about the dihedral angles is that, let's begin with some structure, say hydrogen. If we look at the structure of hydrogen, we can break it down like two hydrogen linked with each other. So there is no chance of formation of any angle or bond. So to define nitrogen geometry of the hydrogen, what you can say is that the two hydrogen molecules, they are separate by a specific distance. When you say the distance, it defines how they are linked and what's the total geometry. So only distance is sufficient for telling the geometry of hydrogen. Now if you take water, the structure of water says somehow like this. Okay, so oxygen attached with 2 hydrogen. Earlier you only say the distance and you get the geometry, right? But here, let's say you know the distance between oxygen and water, this hydrogen and water, and this hydrogen and water. You know that, and you are telling that distances. Let's say this is distance x, this is y. Now my question is, will it give you the clear view of the geometry? The answer is no, because distance is not enough. In this case, you also need to talk about the angle between these 2 hands of hydrogen linked with oxygen. So this angle is required. So for this 3 molecule content, in this case 3 atom content, actually in this case 3, they are linked, you need to use 2 distance and 1 angle to get the geometry correct. On the other hand, if you take hydrogen peroxide, for example, I don't know whether you can see the bottom one, so let me draw it here. So let's say now we take hydrogen peroxide, H2O2. This is another molecule, and 4 different atoms are linked now. And in this case, let's say we have OOH. This is what we have here. Okay, I don't think whether you can see me, but this is how the structure look like, for example, of hydrogen peroxide. Now in this case, as there are 4 points, so this was a 2 point, we only distance is enough. 3 points, 2 distance, 1 angle. Now there are 4 points here. In this case, in this 4 point conditions, we need to talk about 2 different angles. If you think, one is here, another one is here. 2 different angles we need to talk about. Just if you look at the structure of peptide bond here, you will find also 4 different points, N, C alpha, C, N. These are the 4 different regions you will find in between. Okay, N, C alpha, C. In this between, you will find 4 different points, 1, 2, 3, 4. So 4 different points are present. So the similar structure here. So whenever you have 4 different points and the molecule will be in the 3-dimensional shape, in that case there are angles that are formed between planes instead of forming between lines. Because you know, if I draw a line, it will be a 2-dimensional. If I draw a plane, 3-dimensional space. So that's what we are talking about here. So if you look at here, I told you the angle between this 2 line and this 2 line, but we are now calculating this as plane. So just imagine this plane, this line as a plane, imagine this line as another plane. So what we have 2 different planes attached with one another, something like that. This is one plane, this is another plane attached with each other like that. And there will be an angle between these 2 planes, and that angle will be known as the torsional angle or dihedral angle. You can see that thing here. From this, the distance between N, C alpha and C, you will also find that angle, because you know, in this C alpha, hydrogen is attached, R is attached, right? R is the side chain, hydrogen is attached. So if you look at here, you will find that plane, you will find that 4 points, and in each of those sides, this is a 4 point, this is another 4 point. So from that, what you will find, there will be 2 planes creating angles in this bond, 2 more planes creating angles in this bond, right? So there will be 2 different angles, one in the between the N and C alpha, there 2 different planes creating angle known as phi angle, and on the other hand, another between C alpha and C, 2 different planes again creating another angle known as psi. So phi angle in the phi bond, psi angle in the psi bond. Now these angles can change. they can go narrower or broader. This angle can change, so these properties will be changing. If you look at here, those properties will change and the angles are changing, distance are changing, and due to the change of those angles, the rotation quality will also vary. The rotation quality will also change. For example, you say from these two different bonds, the rotations are possible because they are not double bonds, they are single bond, so rotation around that bond is possible. So we have two different angles in which the rotation is possible, because there will be a limit to each of those angles, right? Now, how much those rotations are possible? If it's not involved with any sort of angle formation, or if it's not even a three-dimensional structure, then we can say it can move any place in the same plane. But protein is a three-dimensional structure, and we know it's not only the rotation around the bond, but while it's rotating, the angle is also changing, because due to this angle, the rotation is defined by these angles actually. Okay, so let's say they have a specific angle of plus 60 degree. So once they have the angle of plus 60 degree, the rotation will be possible up to a certain extent only, and only up to that certain extent the protein structure can be stable. Prior to that, or let's say over that angle, or if you change the angle, then only the structure will be changed. So for a specific angle, there will be a specific way of rotation, and that will give a specific structure to the peptide chain. So let's say if the angle is plus 160 degree, they are going to rotate at certain extent. That is going to give them the idea of, or the structure of, beta sheet as a secondary structure. Okay, because these amino acids will try to rotate, they try to interact, they try to form hydrogen bonds between themselves to finally produce a fully functional three-dimensional protein. Now, how they will fold and how those amino acids will interact, that complete thing will depend on the degree of angle, and that will regulate the rotational freedom of all those bonds that will ultimately create whether that amino acid sequence having the specific degree of angles can ultimately form alpha helix or beta sheet or any loop structure. Okay, so that is the idea of torsional angles. So if you know the torsional angles, it will be very important and helpful for us to identify that whether this polypeptide chain is going to form beta helix, beta sheet or alpha helix, or parallel beta sheet or anti-parallel beta sheet, or is it going to form a right-handed helix or a left-handed helix. We can get all these ideas by looking at the torsional angles or dihedral angles, because they are the same things here. Okay, that is all about the dihedral angles. Okay, so as far we talked about the dihedral angle or torsional angle, we saw that there are two different torsional angles formed around this alpha carbon in the peptide chain. One is between the N and alpha carbon, that is known as a Phi bond, where the rotation is possible. Second one is between the alpha carbon and carbonyl carbon, that is known as a Sai bond. So there are two rotations possible. Now just think about, there are so many degree of rotations possible around these two bonds, because if you look at them in three-dimensional space, you will see how those angles are formed. I've already discussed about that. Now let me draw that small section only between this one, nitrogen hydrogen linked alpha carbon. Now remember, alpha carbon have a hydrogen, it also has a R group attached, then we have this. Okay, so this is where they have all those bonds. So let me draw them. This is where they have those Phi bond, this is where they have those, say whatever direction you can, the Sai bond. Okay, so these are the two rotational planes that are possible. Now as you see, there is an R group present here. Now this R group varies, right? We know that R group for different amino acid have different structure. For example, R group for glycine carries very small, the smallest structure, that is only hydrogen, while R group for proline is a complex. and that is the most complex, exactly, and this is the simplest. Okay, so if this R group is a proline, then it's a bulkier ring, a huge bulky ring. So now just imagine there is a bulkier ring placed here, bulky amino acid. Now they try to rotate this part, and there is also another R group present nearby, some R group from another amino acid present nearby, for example. Now as they try to rotate, this bulkier ring will not allow them to rotate properly in all the angles. Why? Because this is a bulkier ring, it will hinder the rotation of the rest of the peptide chain. That is known as steric hindrance or steric problem, or any sort of, like, we can call it as a steric inhibition of those rotation of angles. Okay, so due to these inhibitions or the steric hindrance that is generated due to the R groups that are present there, as a result of that, the rotation is halted. So it depends, the rotation will depend, and the angle of rotation will also depend on the R groups that are going to present and attach with the alpha carbon. Okay, so if a protein contains more proline and proline rich sequences, in that case the degree of rotation allowed will be less in this phi angle, especially this phi regions, it will be less. Okay, and if the carry is more of this glycine, the degree of angle, the freedom of rotation is more, well, for freedom of rotation for proline is less. So now the composition of amino acid determines what kind of rotational angles are permitted for that protein to become a structure called alpha helix or to become a structure called beta sheet. Now it depends on different sort of structures, because for becoming alpha helix a specific angle rotation is required. For becoming beta sheet, a specific angle rotation is required. So if that rotation is possible, then only that peptide structure will form beta sheet. If it's not, then that protein structure will not be able to form the beta sheet. So how could we know by looking at a peptide structure that that peptide is most likely to form helix and not likely to form a sheet, or that structure is going to form a right-handed helix, not a left-handed helix? Or how can we tell by looking at a structure at a linear sequence of amino acid that that amino acid structure is going to form a parallel beta sheet and it is not going to form alpha helix? So that is the question, and we can answer the question by looking at Ramachandran plot. In Ramachandran plot, the discoverer Ramachandran and two or three of his colleagues in India, they discovered the whole process and they put this as in the chart form, that what kind of angles of phi and psi are preferred in what kind of structure in secondary base. So let's look at there. In the Ramachandran plot, there is a graph. The graph is very basic and simple. There are two different, say, x-axis and y-axis in this case. In the x-axis dimension, we can just put it like this fashion. You see here, x will only talk about phi angles. In the y-axis, we will talk about psi angles. Okay. Now in the middle there is 0. In the topmost part plus 180 degree, in the bottommost minus 180 degree of the psi angle. On the x-axis, for the phi angle starts with again minus 180 degree and goes plus 180 degree. So if you look at this graph, it's kind of little unusual in the graph form because it's a quadrant structure will be there in this graph. So in the y we completely have phi angles, in the x completely in the y we have completely psi angles, in the x we have phi angles. So in the y we start with plus 80 here till 0 here and then minus 80 in the bottom, while in x start with minus 80, go 0 here and end with 180 plus. So these are the angle or the rotation possible. Okay, so total rotation of 360 degree, the rotation, because it can circulate completely, that will be 360 degree, but that's how the rotations are for the phi and the psi for two different directions. Okay, clockwise and counterclockwise. Now in this graph they also have a line in the middle. Let me draw it. Okay, so we have four different quadrants. Now according to Ramachandran and his colleagues, the data shows that all those beta sheets that we talk about, they present someplace here in this region. And all the alpha helices that we know of, they tend to present someplace here in this graph. So if you look at here, all the alpha helices are generally present in this area. That means they must carry a higher degree of positive value of the psi and a positive value of the phi. Then only they can be present here. That means all those amino acids that are being involved with to form alpha helices, all of them, they form the alpha helix structure. Those amino acids are allowed to rotate around their both phi and psi angle in positive plus 80, and some range between like 100 to 180 degree positive rotations are allowed. Where in beta sheets, if you look, they are not allowed for more positive phi angle rotation. The phi angle rotation allowed for a beta sheet is ranging from minus 180, like minus 160 to minus 100 and something like that in angle. And again, if you look at little carefully, you will also find that these are for the anti-parallel beta sheets. Parallel beta sheets ranges somewhere here. So though it's an arbitrary graph, you cannot exactly measure the degree of angle that are preferred for different structures. But actually, if you look at the graph which is constructed there, you can find that graph, and there are softwares there. Now if you're dealing with an unknown amino acid sequence, you want to check what will be the folding property of this sequence and what kind of structures this sequence may develop, you can simply check that on Ramachandran plot and put it in the simulation of Ramachandran plot, and it will give you an idea about what is the highly likelihood of that protein to be folded in either helix or beta sheet like structures. And also there are different other structures or secondary structures are possible which are listed in different regions of this graph, but these are the major two things I think is more important to discuss about. That's what we talked about. So this is for the alpha helix, and this is anti-parallel beta sheet, and this is for the parallel beta sheet. So this in a sense are the total idea of dihedral angles and the role of dihedral angles to find out what kind of protein structures it will ultimately help to form with the help of Ramachandran plot. Okay, so remember, Ramachandran plot's idea is very simple. It's based on the angles with which phi and psi bonds start to rotate, and those amino acids start to rotate around the phi and psi bonds, and the degree of freedom they have to rotate. Okay, the idea of the freedom to rotate around those bonds and the angle that is permitted to form a specific secondary structure like beta sheet or alpha helix, that thing is measured with Ramachandran plot. So if we are telling that for becoming a beta sheet you need to have the phi and psi angle ratio like this, if it has a different phi and psi angle ratio, that amino acid structure will not try to form beta sheet. They will go to form another type of secondary structure. Remember, because it depends on when the structure of the protein, that when it's going to form alpha helix or going to form the beta sheet. Another thing that regulates how these amino acids are going to rotate around those phi and psi bonds is the R group. If the R group is proline, in that case the freedom of rotation is restricted because the proline is a bulkier group. If the R group is a glycine, which only carries this glycine R group is H, because glycine only carries a very small group, so the freedom of rotation for the glycine is higher here. So this is the difference between and this is the idea between the Ramachandran plot and the protein structure, and why Ramachandran plot is important. So I hope you understand this video. If you like this video, please hit the like button, subscribe to my channel to get more videos like that, and share this video with your friends. Thank you.